What is food testing? Food testing is a process to analyze a food and the content of the food. Food testing will provide the information about characteristics of the food, such as structure, composition, and the physical-chemical properties. Other than that, it is also done to test the quality of the product for quality control and food inspection. This process is the last step of food manufacturing because it is really important to determine the safety of the food. Food testing can be done in 6 methods, such as analytical chemistry testing, sensory testing, nutrition analysis, microbiology testing, chemical contaminant testing, and the last one is drug residue testing. Food analysis use powerful instrumental techniques used in labs to increase the specificity, precision, analytical accuracy, and detection limit in food testing. The food sample that will be used for this assignment is from bee, which is honey. Honey is composed of carbohydrate.
In theory, high-performance liquid chromatography or high-pressure liquid chromatography (HPLC) is a chromatographic method that is used to separate a mixture of compounds in analytical chemistry and biochemistry so as to identify, quantify, or purify the individual components of the mixture. In very small amount, the sample mixture to be separated and tested is sent into a stream of mobile feeds, percolating via a column. There are different types of columns available with solvent of varying particle size in surface. The mixture moves through the column at varying velocities and interact with the solvent, also known as the stationary phase. The velocity of each component in the mixture depends on its chemical nature, the nature of the column, and the composition of the mobile feeds. The HPLC partitioning process is quite similar to the liquid-liquid extraction process, except that the former is a continuous process, unlike the latter which is a stepwise process. It is recommended that trial partitioning process be performed to determine the exact HPLC method that would provide adequate separation.
The methodology for HPLC consists of an infusion system, degassing device, gradient elution device, sampling system, separation system, detector, and data processing system. The infusion system is used to apply the suitable pressure based on the output of honey. Degassing is used to eliminate bubbles entering the detector, while the gradient elution program is to control the composition of mobile phase within the analysis of honey. Sampling system is required to have good repeatability to ensure center sampling and that the flow rate of chromatography system remains small. Later, the separation system includes chromatographic column and a column oven to further the analysis of honey. Then a detector is used to monitor the composition separation of the component in honey sample. And finally, the data processing system processes the test data correctly, showing the results of each component that is present in the honey sample.
For this analysis, we have to manage two structural different compounds. Carbohydrates are very polar compounds, while HMF is an organic compound that acts hybrid and a polar moiety. Several conditions may be investigated, but a fast and convenience analysis have been developed using the porous gravitic columns that allow the separations of both polar and apolar compounds within a single reverse phase types gradients. HPLC column will allow internal diameter of odor to decrease the solvent consumptions. The ELST used for this experiment is ZDAC-LC, a universal detector capable of detecting all solvent with less volatile than the mobile phase. This detector allow very straightforward quantifications of carbohydrate thanks to 5 decades of dynamic range, accommodating easy with multiple concentrations level in the same samples.
In summary, the standard methods of analysis required by legislation and expressed by the HPLC that has shown small variations. However, more advanced techniques and methods have also been used in honey characterizations with the analysis of minority components and in the research of adulterescence parameters. The use of state-of-the-art procedures and non-destructive online methodologies are nowadays becoming increasingly important in the food industry. Plenty of laboratories that related to big products are already using modern technology to study the components present in honeys.